Welcome to Lecture 16, where we will take a look at meiosis, or cell division in gamete cells. There are a few base definitions that will be important through this chapter. First is genetics, which refers to the study of heredity, or how genes are inherited from parent to offspring. Heredity refers to the passing on or transmission of traits from one generation to the next. Variation occurs in the process of sexual reproduction, and this refers to the fact that offspring are not considered to be genetically identical to either parent or to each other. Individuals have unique combinations of parental genes that adds to variability in the genome. Asexual reproduction occurs when a single organism gives rise to their own offspring. This typically leads to the production of clones or genetically identical individuals. The study of heredity and genetics can be done through the use of a life cycle. This provides the generation-to-generation sequence or genetic stages in the reproductive process. If we take a look at the human life cycle, we know from the previous lecture that somatic cells are considered to be diploid because they have two copies of each chromosome. In somatic cells in humans, we have 46 chromosomes, 23 originated from our mother and 23 originated from our father. Hence, we are considered to be diploid with two copies. We can refer to that diploid state as 2N. Gametes, on the other hand, or germ cells, such as the sperm and egg in humans, are considered to be haploid, meaning that they only have one copy for each chromosome. We can term that 1N. As stated previously, humans have 23 pairs of chromosomes, 23 from the mother and 23 from the father. As a result, our N value, or number of chromosomes, is 23. So in the diploid state, the genome in our somatic cells, we refer to that as 2N, where 2N equals 46. This gives our total of 46 chromosomes. When we are considering the human life cycle, the fusion of two gametes in the haploid state results in a 2N zygote. So this means that when gametes undergo fusion to produce a zygote, we have the return of the diploid state. A karyotype can be very important in looking at the genetic material of individuals. Karyotypes are typically arranging the chromosomes according to length, longest to shortest, keeping the sex chromosomes to the last position on the karyotype chart. We call our chromosome pairs homologous. This is because these chromosomes represent the same traits. However, the genetic material making up those traits isn't necessarily identical. As we saw in mitosis, we have two sister chromatids that contain genetically identical information. When we're dealing with our chromosome pairs, these are considered to be different. Remember, one originated from your mom and one originated from your dad. So though they represent the same traits, they're not considered to be genetically identical to each other. So we cannot call these identical sister chromatids. Instead, we call these homologous chromosomes. This is an example of a typical karyotype. We can see here that chromosome 1 is the longest, arranged all the way to chromosome 22 being longest to shortest. We can also see the 23rd chromosome pair is our sex chromosomes, and in this case we have an X and a Y, representative of a male. Within the human genome, we have 22 pairs of autosomes. These are considered to be non-sex chromosomes and are represented in the karyotype on the previous slide in the chromosome position 1 to 22. In humans, we also have one pair of sex chromosomes, which are your X and Y chromosomes. So in total, we have 23 pairs of chromosomes. Biologically speaking, we have the differentiation between females and males, where females have double X chromosomes and males are present with the X and Y chromosome. Because the X chromosome is considered to be present in both males and females, it is considered to be an essential chromosome. However, the Y is not considered to be essential to life. The X chromosome is also very large in proportion to the Y chromosome. This is because it does contain a wide variety of important genes outside of just sex determination. Of course, we know that sex and gender is much more complicated than this. However, it is just simplified in this form for this course. We also take a look at some of the genetic abnormalities that can result from differences in the sex chromosomes. Meiosis refers to the process of making those haploid gametes from diploid precursors. So special pregametic cells are going to undergo meiosis in order to enter into the haploid state. Fertilization, can also be termed syngamy, occurs when the gametes fuse together. In humans, this results in the reestablishment of the diploid state. You have a sperm in the haploid state joining with an egg also in the haploid state. This genome is going to combine to form the zygote, which is then going to restore the diploid or 2N state. The resultant offspring will then have a unique combination of both maternal and paternal chromosomes. When species reproduce sexually, they have to alternate between the haploid and diploid stages in order to exchange genetic information and to increase genetic variability of their offspring. In animal cells, the haploid stage is considered to be very short. Consider our own life cycle. Our haploid state only exists in the formation of the gametes. All of the rest of our somatic cells and organisms are considered to be in the diploid state. However, many fungi are actually the opposite, such that the adults are considered to be haploid, and when the gametes fuse, a diploid zygote is created quickly that undergoes meiosis to enter back into the haploid state. In plants, both of these stages, haploid and diploid state, can be very long, and some can even be multicellular. This means that there are a wide variety of strategies that different organisms can put into place to increase their genetic variability and to reproduce. This slide depicts the different life strategies that can be present in eukaryotic organisms. If we take a look at the first cycle, which is animals, this is very similar to our own life cycle, where we've got a zygote that is diploid, 2N, and those cells will undergo mitosis to produce your multicellular organism that will also be considered diploid. This is equivalent to our somatic cells. Some of these cells, however, in humans are pregametic cells, will undergo meiosis to form a haploid gamete. These gametes will then fuse during sexual reproduction to lead to the production to a zygote. Plants and some algae, however, have a different strategy. They can undergo fertilization to once again form a zygote, which will undergo mitosis to form a diploid multicellular organism. In this case, we call the diploid form the sporophyte. The sporophyte can have specialized cells that will undergo meiosis to form spores, which is what we see in the diagram. However, some of these spores will undergo mitosis to form a haploid organism called a gametophyte. This gametophyte then has the ability to produce gametes. So specialized cells within the gametophyte will be produced into gametes. These gametes will then undergo sexual reproduction, which will then undergo fertilization to form the diploid multicellular organism or the sporophyte. So we can see that there's two different life forms, haploid multicellular as well as diploid multicellular, that can be utilized in this life strategy. Some fungi and protists can undergo a different life cycle strategy. In this form here, the diploid state is considered to be a very short phase in their life cycle. So their gametes will undergo fertilization to form the zygote. However, the zygote will undergo meiosis to form a diploid or haploid cell. This haploid cell will undergo mitosis to then form a haploid organism. This haploid organism can also undergo mitosis to then produce other gametes. If sexual reproduction is necessary, these gametes will then fuse to once again form the zygote. However, the zygote will quickly undergo meiosis to return to the haploid state. In summary, meiosis is a very important process because this allows for the organism to reduce the chromosome number from a diploid to a haploid state. It does so by separating out the homologous chromosomes. Remember, homologous chromosomes represent the similar chromosomes that you inherited, one from your mother and one from your father, one maternal and one paternal, that represent the same genetic information. Even though they're not genetically identical, they carry the same traits. It is also important to remember that meiosis itself is preceded by the various stages of the cell cycle, meaning that we still have a gap phase, we still have an S phase where DNA is synthesized, and we still have a G2 phase. However, instead of undergoing mitosis, these cells are going to undergo meiosis, where we're going to divide out that genetic information twice. When you have that second round of cell division, there is no S phase in between that. This allows for the separation of the genetic material to be able to go from the diploid state to the haploid state. As a result, when we consider meiosis, we have two separate meiosis phases, meiosis 1, which refers to the first division, and meiosis 2. When we consider what we are separating in each of these phases of meiosis, it is important to understand that when we're dealing with meiosis 1, this is when our homologous chromosomes are separated. This means that we are separating out, say, chromosome 1, we're separating out our paternal and our maternal copy. This means that at the end of meiosis 1, we have essentially reduced our genetic information from the diploid state to the haploid state. We've gone from 2N to 1N at the end of meiosis 1. When we consider meiosis 2, this is when we're going to be separating out the sister chromatids. Because remember, when we have our maternal and paternal chromosomes, both of those are undergoing S phase, which means that for our paternal chromosomes, we're going to have an original and copy. And for our maternal chromosome 1, we are also going to have an original and copy. So that means that in meiosis 1, we're simply separating out the maternal chromatids from the paternal chromatids. And in meiosis 2, we're going to actually pull apart those chromatids. This is going to overall result in the production of 4 genetically distinct daughter cells, and each of these daughter cells are going to be haploid, containing only one version of the gene. This slide helps to depict the difference between those two divisions. Remember, we still have interphase occurring, so we still have the 2 growth phases and we still have the S phase where the DNA is going to be synthesized. This is what we see in the first part of this diagram. We have our homologous pairs of chromosomes. Let's say this is chromosome 1. We have a copy from our dad's, a paternal copy. Let's say that is the chromosome in blue. And we also have a maternal version of the gene, which is in red. Once this cell undergoes S phase, that DNA, both of those, will have to be copied. And this is what we see in the second cell underneath. We now have an original and copy for our dad's paternal genes, and we also have an original and copy for the maternal ones. These are considered to be sister chromatids. We have an original and we have a copy. Now when we undergo meiosis 1, we are going to separate out those homologous chromosomes, and this is what you see. You can see that at the end of meiosis 1, we have our dad's original and copy in one cell, and our mom's original and copy in the other cell. So at the end of meiosis 1, we have produced 2 daughter cells. Each of these are considered to be haploid because they only contain either the paternal or maternal DNA. They don't contain both. We are then going to enter into meiosis 2. Each one of these cells, the maternal and paternal copy cell, are going to undergo a secondary round of division. In the secondary round of division, or meiosis 2, This is where we have the separation of chromatids. This is why we can see that in meiosis 1, if we take a look at the paternal cell, the one that has the two paternal chromatids, when this cell undergoes meiosis 2, those chromatids are going to be pulled apart. As a result, you're going to get the production of two haploid cells. Similarly, in the mother's copy, the copy with the red genome, the red chromosomes, those are also going to be separated out. Those chromatids are going to be pulled apart in meiosis 2 and also result in the production of two additional haploid cells. As a result of this process, we have gone from one diploid parent cell to the production of four haploid genetically distinct gamete cells. This is the process of meiosis.